Start off with a question. Raise your hand if you have food allergies. Okay, look around. Raise your hand if you know someone who has food allergies. Okay, so that's a lot of people. So around 16 million people in America have food allergies, and that's a lot of people. And right now, there's no cure for allergies, and they don't really know why you have allergies. So some people are born with them, or you could develop them later in life. So first off, what is an allergy? So it's when your immune system responds to something in your environment. So it could be pollen, it could be food, and it could even be medicine. So allergic reactions, you have certain symptoms, which could be hives, itchy throat, difficulty breathing, and the worst one's an anaphylactic shock. And when you have an anaphylactic shock, your airways start to close up and it's really hard to breathe. And if it's not treated quickly, you can die. So right now, the treatment they have for allergies is an EpiPen, which that helps lessen the symptoms and helps loosen up your airways so you can breathe better, and then also Benadryl, which you take if you have like a hive. So the article title was Genome-Wide Association Study Identifies Peanut Allergy Specific Loci and Evidence of Epigenetic Mediation in U.S. Children. So to put that in simple terms, there was a study that located a specific area on a chromosome that people with peanut allergies had. So to go over some quick definitions, a genome is the complete set of genetic material in a cell or organism. You have 23 sets of chromosomes in your body, as you can see here, and they all contain genetic information which code for specific proteins, and then their genes, which are made up of a line of nucleotides, and they code for specific proteins, and they get passed down from your parents to the offspring. So then loci, there's a specific location on your chromosome, and those are loci, and they can potentially identify genes in that location. And then methylation, which will come up a little bit later, is when you add a methyl group to the DNA without changing its sequence. And as you can see here, it's the methyl group added to cytosine, and it represses gene transcription. And then SNP stands for a single nucleotide polymorphism, which is a change in a single nucleotide in DNA. As you can see here, AT is now GC. And then intergenetic SNP is when there's an SNP in between genes, so it shouldn't affect gene transcription. So then the study was broken up into 3 parts. Discovery, which is where they tested individuals for food allergies, and they also tested if there was an association in their genes to food allergies. And stage 2 was replication, where they replicated those specific areas that they found associated with food allergies to confirm that it wasn't just a coincidence that they found that association. And then in stage 3, it was methylation mediation, where they compared the SNPs and DNA, where they added where they methylized DNA to see if there was still that association between the SNP and the peanut allergies. So in stage 1, they did the testing to see if the individuals had food allergies, and they tested individuals of European and non-European ancestry. And then they also tested for general food allergies, which are wheat, nuts, shellfish, the main food allergies. And then they also narrowed it down to peanut, egg, and milk allergies, which are the 3 most common food allergies. So individuals of European ancestry, they found a strong association with peanut allergies in those people of European ancestry in chromosome 6. And then they didn't find any associations with individuals of non-European ancestry. So then they also tested specifically for milk or egg allergies, and they didn't find any significant association with those SNPs and those allergies. And then they tested those 2 specific regions that they found associated with peanut allergies for milk and egg allergies, and they found no association. So they really narrowed down their focus to people with peanut allergies. So this graph shows, or this chart shows here are the 23 chromosomes, and here there's the peak on chromosome 6 because it's significantly associated with peanut allergies. And then they also, down here, they provided the genotypes and the imputated SNPs, which mean they also tested for alternative forms of the genes, and there's still that significant peak on chromosome 6. Any questions? So then in stage 2, they did replication. So they wanted to confirm that it wasn't just from the data that they gathered, that there was that significant association in those 2 spots. So they replicated the 2 most significant SNPs to see if there was still that association. They confirmed there is. So then here's another chart, shows this is chromosome 6, and these are the different regions on it. And up here are those 2 specific points that were most associated with peanut allergies. And then stage 3 was methylation mediation, where they added the methyl group to the DNA to see if it would, so when they added it, it represses gene transcription. It expresses the gene from showing. So when they tested it, they did see that once they added that methyl group, there was an effect on the association between SNPs and peanut allergies.
Sorry, can you tell us a little bit more about that? So they added it in was this like in a cell line? Like what were they? Tell us a little bit more about this experiment.
Yeah, so they added the methyl group onto the DNA.
At this specific loci?
Yes.
Okay.
And it represses the gene transcription, so they wanted to see if people who had the peanut allergies, if that one specific gene that's associated with the peanut allergy wasn't expressed, if they would still have a reaction.
What gene is this SNP in?
It's in the HLA class, which is in that class, a lot of the genes in it code for the immune system. Okay, so the results. So in stage 1, they identified and genotyped individuals with European and non-European ancestry. They noted down their food allergies, and they narrowed their focus in stage 2 and found those 2 specific locations on chromosome 6 that had a significant association with peanut allergies. And then in stage 3, they concluded that even though they added the methyl group to the DNA, there's still that significant association with peanut allergies. And then they also noted that DNA, when it's methylized, might mediate the risk of peanut allergies. So then, why does this study matter? It matters because this was the first study that they really looked into the relationship between methylized DNA and food allergies, and it could lead to potential treatment for people with peanut allergies. And then also when children are born, they can do a DNA test on them and see if they have those significant areas on chromosome 6 to see if they have a peanut allergy, because right now the only way to know is if the child eats peanuts and if they have a reaction, or they do a skin test. So when the baby's born, they can immediately know and do further testing to know if they have a reaction. Any more questions?
Do you know why they separated between people with European ancestry and non-European ancestry?
The study just said they wanted to see if there was a difference, and they did find that there was, because there was only that significant peak of people with European ancestry.
Just like interesting tidbit to add on that. We always, well, my family at least, likes to talk about the fact that in Israel, there's a much lower peanut allergy rate than in a lot of other places in the world, and scientists had previously thought that it was like, because there's a very popular junk food that has peanuts in it that you feed kids at a very young age. So I thought it was kind of interesting because this study sort of points that like it's really more of an ancestral inheritance kind of thing.
So in stage 3, when you talk about how they methylized the DNA, was that they took someone who was allergic to peanuts and then they methylized tested to see the effects?
Yes.
In a person? Like, that was kind of my question too. Like, they weren't testing this on
Yeah.
people, right? Were they just looking at correlation, like people who were methylated versus ones who weren't? Or they were actually like changing the methyl?
Great.
The study didn't really go into that much